Before we start new discussion for today, so I would just like any one of you to remember, or every one of you to remember our previous topic. What is that all about, class? What is that all about? What we've talked previously, because it's very important that
There,
we recall our previous topic, because that's gonna be grounded to our topic for today. Can somebody recall?
sir,
Yes,
cell
Jaila.
division po. And
Oh,
dun po sa parang may PMAT po.
okay, so particularly we're talking there about mitotic cell division as part of the cell cycle. Anyway, our another topic for today is also another part of cell division or cell cycle. However, it has different purpose. Okay, so remember, class, that that is mitosis. Okay, so mitosis, the purpose of that is what? What is the purpose of mitosis in our body? Hmm. What is the purpose of mitosis in our cell body or in our body? Okay, so for cell division, so what is that for? The cell division. What is that for? Yung cell division. What's the end goal of having cell division in our body? Anyone? I observe you barely remember our previous topic. Okay, very good for that, Jaila. So for wound healing, okay, so that's one purpose of having mitosis. Okay, so wound healing, cell growth, cell reproduction, okay, so growth of our body, cell repair, tissue repair. So that's the purpose of mitosis. Okay, so by this time I'm gonna share you my slide. So this will be about what? About another type of cell division. You call this cell division as meiosis rather, okay? In addition to the mitosis we've previously discussed, okay? So meiosis has a different purpose compared to mitosis, okay? So how is that so? We will know after this discussion kung ano ba yung purpose ng meiosis, okay? So it's pronounced as meiosis, okay? So in this discussion, the main objective is for us to differentiate meiosis from mitosis. Though they have some similarities, but we will focus on their what do you call this? Differences. Okay? So I hope you can see my slide clearly. Am I? Or am I sharing the slide
Yes
already?
po.
Okay, very good. Okay, so we'll proceed with the discussion on meiosis. But before that, let's take a look of our objectives for today. So number one, can somebody read here? Number one is, walang nagbabasa, ako na nga lang. So compare mitosis and meiosis and their role in cell cycle. So yan, we are going to compare and contrast them. Okay? And also, identify the goal of meiosis in cell cycle. So the second objective that we have today is to determine how their phases contribute to the reproduction of sex cells. Okay? Again, determine how their phases, kasi as compared to mitosis, it also contains PMAT. However, it's a different form. Contribute to the reproduction of sex cells. The third one is to explain the role of meiosis in sexual reproduction. Okay? So, it's more of sex characteristics yung ating i-discuss ngayon. Okay po? So, let's proceed with the presentation. Okay? When we speak of meiosis class, it's the type of cell division that produces haploid. Okay? When we speak class of haploid, it contains half the number of chromosomes. From 46 that we have in our body, in our normal cell or somatic cell, so it only contains 23 chromosomes. Okay? So, and these 23 chromosomes or haploid sex cells that we have in our body are gametes or sex cells. What are these gametes or sex cells? They are synonyms. Gametes and sex cells. They are the egg cell and the sperm cell. Okay? So, female and male respectively. Okay? So, these are for males. Sperm cells are haploid cells that resulted from meiosis. So, if I asked you what's gonna be, what cell type or what are the cells in your body that are haploid? So you might say, sir, these are the sperm and egg cell for male and female respectively. Okay? So I hope you're getting what I mean with this one. Okay? So next. Okay, so meiosis is divided into 2 rounds. Okay? Kung kanina, kung sa, what do you call this, kung sa mitosis, it only contains 1 round or the PMAT round. So, itong meiosis, meron siyang 2 rounds. So, we have meiosis 1 and meiosis 2. So, later on, we'll know bakit siya reductional, yung meiosis 1, and bakit siya equational, yung meiosis 2. Okay? So, yan. Okay, so let's proceed. Okay, so one thing unique in meiosis 1, so sa meiosis muna tayo, meiosis 1, kasi 2 rounds siya. So, is the presence of synapses. Okay, when we speak of synapses, you can also call this one as, what do you call this? Crossing over. Okay, crossing over of segment of a particular chromosome. Okay? In meiosis class, remember class that there should be a presence of homologous chromosome. Di ba? In a previous discussion that we have, when we speak of homologous chromosomes, these are chromosomes that are identical in appearance. Okay? Identical in appearance in such a way that it has the same length, the same sets of genes. However, they are different in a case of different forms of genes like alleles. Okay? So, these are homologous chromosomes. Okay? So, remember that homologous chromosomes, one comes from your father, the other came from your mother. Okay? So, synapses is the event in meiosis 1 that involves close association between homologous chromosomes. So, how that synapses happen, or how does that synapses happen? Okay, so there should be overlapping. Okay, you call that synapses the overlapping of homologous chromosome. Okay, so nagkakaroon siya ng overlapping during the prophase 1 or the meiosis 1. Kasi class, in meiosis 1, we have PMAT 1. In meiosis 2, we have PMAT 2. So, there are 2 rounds of, what do you call this, division. Okay? So, when this happens, so we can form here chiasma. Okay? Chiasma is the portion of homologous chromosomes when 2 homologous chromosomes meet. Okay pa? So, you call this class as bivalent or tetrad. Okay? Bivalent because there are 2 homologous chromosomes overlapping. Okay? Tetrad because there are tetrad. Okay? 4 involved sister chromatids in this case. Okay? So, that are overlapping. Okay, so one thing that you should observe under synapses is that there should be an exchange. Okay? Exchange can be termed also as recombine or recombinance. Okay? There should be an exchange of a segment between non-sister chromatids. Remember class that, wait lang, may I use the highlighter? Okay. So, what do you call this? There should be an exchange between non-sister chromatids. Exchange of what? Exchange of segment or portion. Okay? Remember that this particular homologous chromosome, this segment is a sister chromatid of this segment. Okay? So, the blue ones are sister chromatids and the red ones are also sister chromatids. Okay? There should be an exchange between two non-sister chromatids, the red and blue. Okay? When that happens, the recombinants are formed. So these are the recombinants. A portion with a blue and a portion with a red. Same thing with the other, a portion with a red with a portion with a blue. Okay? You call this class as daughter chromosomes. Okay? After recombination happens. Okay? So for diploid organisms, synapses involves the formation of tetrad. This is for diploid. Okay? So formation of tetrad. Okay? So next, crossing over or genetic recombination. This is what I've already said. So there should be a crossing over. Di ba in a basketball, pag sinabing crossing over, so you pass along with your opponent. Okay? So in this case, the opponents are the non-sister chromatids. Okay? So there should be a crossing over or genetic recombination. Okay? So genetic recombination means exchange. Okay? To simplify the term. Okay, so this is the exchange of genetic material between the members of homologous pair. In this case, the red and the blue ones. Okay po? So I hope you're getting this sense. So later on, we're just going to focus on the process. Okay? Cellular process of meiosis. Okay? Mamaya na natin identify kung paano ba siya nag-work, ano ba yung purpose niya. Okay? So, yan. Later na lang. Okay, so as I said, in meiosis 1, there are PMAT1. That's why we have prophase 1, prophase 2. Okay? We also meiosis 2. That's why we have prophase 2, meiosis 2. Prophase 2, metaphase 2, telophase 2. Dito naman, pag meiosis 1, lahat lang 1. Prophase 1, metaphase 1, anaphase 1, telophase 1. Okay? So what are the features? When we speak of features, these are things you can observe during this stage. Okay? Prophase 1, under this one. So we have 3. So nuclear membrane begins to dissociate. Okay? So nuclear membrane and nucleolus, diba? Remember the nuclear membrane? That membrane encloses the chromosome or the DNA inside the cell, and the nucleolus, it's inside the nuclear membrane, responsible for ribosome production. These two disintegrates. When we speak of disintegrates, it breaks down in a way that's not visible. Okay? That's not visible during the cell cycle or during the meiosis. Okay? So, for chromosomes, it condenses. Okay? So, it begins to condense. Same thing with the mitosis. Okay? So, remember class we are talking about the meiosis stage. Okay? And the meiosis is part of a cell cycle. Okay? And a cell cycle contains interphase where G1, S, and G2 phase happen. So, we are assuming that here already that interphase already happened. Okay? So, we assume that interphase already happened in this case. So, we are on the sub-stage of cellular division or cell cycle, and that is the meiosis. Okay? And I am just reminding you that meiosis contains 2 sub-stages or 2 rounds of division. So, we have meiosis 1 and meiosis 2. Okay? So, same lang yung pattern, PMAT, pero dalawa lang sa meiosis. Okay? So, next thing, Or next feature of prophase is synapses and crossing over of homologous chromosome. Okay, so that's under prophase 1. Okay, so here is the centrosome. It also begins to move to the opposite poles, forming synchronously with the spindle fibers. You call this class as spindle fibers. Okay? So centrioles are two sets of microtubules. Centrosomes are the whole set, okay, of centrioles. Okay? So chiasma, so yung kanina, it's where the intersection of recombinants happen. Okay, so yan, homologous chromosome, nuclear envelope fragments. So in this case, fragments pa lang. Still visible pa din yung fragments ng nuclear envelope. Okay? So, yan. Okay, for metaphase 1, okay, so that's after prophase 1, homologous pairs at the metaphase plate. Okay, so it aligns at the equatorial plate. Okay, so imagine that a cell is just Earth that has an equator. So homologous pairs, okay, which are after recombination, aligned at the middle. Okay, so in this case, pairs sila. When we speak of pair, kung sa mitosis, isa lang yan na chromosome na duplicated chromatids. Na duplicated chromatids. Okay? Sa mitosis. Sa meiosis, hindi. They are homologous chromosomes. Okay? Na magkatabi. Okay? Or they are overlapping towards each other. Okay? So, naka-align na siya sa metaphase plate. Okay? One with a recombinants, meaning may red and blue. One, what I mean is, two chromatids here are recombinants, meaning two with red and blue, and the other two is red. The other one is red, the other one is blue. Okay? So, later on, I can give you a picture of how closely you can see metaphase here. Okay? So, yan, homologous, okay, are oriented from each other at opposite poles. Okay? So, and the spindle fibers are attached to their kinetochore. So, meaning, one of the homologous are oriented in the opposite pole here. Okay, it has a kinetochores which attaches the spindle fibers, and the other on the other side of the plate. Okay, and the other homologous. Okay, same thing with the other sets. Okay, so here in this case, pinapakitaan lang tayo ng tatlo. 1, 2, 3. 3 pairs. But in reality, it should be 23 pairs. Or 46 rather. Okay ba? So, here is the microtubule extending from the centrosome towards the homologous. Okay? So, next for anaphase 1. Okay, for anaphase 1, this is after just the metaphase 1. Spindle fibers pull the homologous chromosomes instead of sister chromatids in mitosis. Diba? In mitosis, it breaks down the centrosome. Okay? So, that's it breaks down the centromere rather. That's the center of two sister chromatids. Replicated sister chromatids. It breaks down the centromere, pulling sister chromatids apart. In this case, not the sister chromatids, but the homologous chromosomes. So, it pulls the homologous chromosomes apart. Tignan nyo yung illustration. One of the sister chromatid is blue. The other sister chromatid is blue with a red portion because it it's after the process of recombination or crossing over then. Okay, same thing with the other pair. So it pulls apart homologous chromosome. Okay, chromosomes are still duplicated. Why? Because nag-pairing kasi yung homologous chromosome. Okay, yun yung unang pinag-pull apart. Okay, so let's continue. Okay, so next, after that telophase, same thing with mitosis. These are the features. There is the presence of cytokinesis, which occurs alongside with the telophase 1, meaning separation of the whole content of the cell, including the cytoplasm and cell membrane, okay, by pinching outward towards the inside. Okay, members of the duplicated homologous chromosomes reaching the opposite poles. Okay, okay, haploid daughter cells are formed. Okay, haploid daughter cells are formed. Okay, so in this case, actually hindi pa siya haploid, just because we are just in the meiosis 1. Okay, nagiging haploid siya. Diploid pa siya kasi duplicated pa siya. Okay, duplicated pa siya. So, there should be a correction with this one. So, duplicated pa siya. So, kaya siya diploid pa siya. Okay? So, next, we're going to talk about meiosis 2. So, meiosis 2 in this case, immediately follows after first mitotic division. Paano 'to nangyayari? Because wala nang interface or cell growth or pag sinabing cell growth, yung G1, wala nang S na duplication of DNA, wala nang G2. It follows immediately without the presence of interface. Okay? So, remember that there is no interface between meiosis 1 and meiosis 2. Okay, so the second mitotic division is equational. Okay? Meaning there is no further reduction in the number of chromosome. Okay? There is no further reduction in the number of chromosome. It is not preceded by DNA replication. So as I said, there is no interface in between. It begins with 2 haploid daughter cells of meiosis 1. Okay, so yan. So the process is similar to mitosis, which involves the separation of sister chromatids. Okay? So in this case, the duplicated ones, so sabi ko nga dalawa siya. So in meiosis 2, the sister chromatids, okay, which are bonded by centromere, will fall apart or will separate siya. Okay? So in this case, I'm gonna show you this one. Okay, so for prophase 2, okay, so yan na eh. Basta ganito, prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis. Okay? So if you observe the illustration, okay, meron tayo ditong mga captions. So in this case, a new spindle forms around the chromosome. So same thing, spindle forms around the chromosome. So yan. Okay, so in metaphase 2, it lines up, okay, the sister chromatids. Actually, they are not already sister chromatids because it undergoes cell division. What do you call this? Crossing over already. So they are not classified as sister chromatids, but we can say already that's duplicated chromatids. Okay? So basta dalawang chromatids that are attached. So it aligns at the equatorial plate. Next, it separates, it breaks down the centromere and separates the sister chromatids. The chromatids apart from each other towards the opposite pole. Same thing with mitosis. And here, in this case, yan, separated. Okay? So, in this case, parehas na siya ng mitosis. Kasi, it breaks the sister chromatids apart or the chromatids apart towards the opposite poles. However, the difference is the content of the DNA or chromosome. One has a recombinant, the other one is pure. Okay po? So, yan. So, these are the, what do you call this? comparison. Mitosis and meiosis are differentiated from each other by using the following parameters. Number one, number of daughter cells and cytokinesis involved, because in daughter cells, we have 4 actually. Kasi from 1, it divides into 2 during meiosis 1. From 2, it divides into 4 during meiosis 2. So number of daughter cells in meiosis, in mitosis, we have 2. In meiosis, we have 4 actually. Okay, so yan. Cytokinesis involved in 2 rounds. So for mitosis, it's only 1 round. For meiosis, we have 2 rounds. Chromosome number and of daughter cells. Okay, for chromosome number of mitosis, we have 46. 46. Okay, for meiosis, we have 23 because it's haploid. So types of cell involved. Okay, so here in mitosis, typical somatic cell. Okay, brain cell, blood. Okay, so typical cell. Okay, for meiosis, the cell involved are the cells in your gonads. You call this as, what do you call this? Gonadal cells, or sperm or egg producing cells. Okay, so that can be found in your gonads. Okay, so types of cell involved. Next, presence of unique events such as synapses and crossing over of homologous chromosomes for meiosis, and there is none of this in mitosis. Okay pa? So I hope you're okay. So before we come to this one, so I just want you to watch the video that I'm going to present. Okay, this one is a very good video that will explain meiosis further. Okay pa? So I want you to listen and attentively. Okay? Let me play this one.
Do exactly like their parents, or why? Have you ever wondered why children do not look exactly like their parents, or why some siblings look so different from each other? The answer lies in the process of meiosis, which generates the gametes for reproduction. Meiosis is the process by which haploid cells are produced from a diploid cell.
Okay.
For this to occur, the chromosomes must be correctly sorted and distributed in a manner to create genetically unique cells with half the number of chromosomes as the original cell. Meiosis occurs in special cells called germ cells within the gonads of males and females. For the successful reduction in the number of chromosomes to occur in the new haploid daughter cells, 2 rounds of divisions are necessary. These divisions are termed meiosis 1 and meiosis 2. Although the processes of mitosis and meiosis look similar, there are a few major differences. One obvious difference is that mitosis results in 2 diploid daughter cells, and meiosis results in 4 haploid cells. As in mitosis, meiosis begins after a cell has successfully completed the G1, S, and G2 stages of interphase. During the S phase of interphase, the DNA is replicated, producing 2 copies of each chromosome called sister chromatids. The paired centrioles in the cytoplasm duplicate and begin extending microtubules that will form the meiotic spindle. Sister chromatids remain attached at the centromere and condense as the cell enters prophase 1 of meiosis. Up to this point, the cell looks similar to mitosis, but 2 events occur in meiosis that do not occur in mitosis and lead to genetic diversity. The first event occurs during prophase 1, when homologous pairs of sister chromatids lie side by side in a process called synapses, forming a tetrad or bivalent. The homologous chromosomes share similar, but not necessarily identical genes. Once this structure is formed, the second event, called crossing over, can occur. During crossing over, a physical exchange between chromosome segments of non-sister chromatids occurs, increasing genetic diversity. Prophase 1 concludes with the fragmentation of the nuclear envelope as the duplicated centriole pairs move to opposite poles of the cell. As they move, the centrioles extend spindle fibers, forming the meiotic spindle. In prometaphase 1, with the paired centrioles in place, the meiotic spindle is fully formed. The sister chromatids attach to the spindle fibers by their kinetochores. Here, another key difference between mitosis and meiosis occurs. Because of synapses and crossing over in meiosis, homologous chromosomes remain aligned, so that a pair of sister chromatids is attached to only one pole by the kinetochore microtubules.
So, isa lang.
In mitosis, each sister chromatid is attached to a spindle fiber. The fibers originate from opposite poles. During metaphase 1, bivalents randomly align along the metaphase plate. Due to independent assortment, this alignment is random and adds to genetic diversity. In anaphase 1, the homologous chromosomes separate and move toward opposite poles. Meiosis 1 ends with telophase 1, when the chromosomes decondense and the nuclear envelope reforms. Cytokinesis separates cytoplasmic material, and the two daughter cells are separated by a cleavage furrow. Since the final product of meiosis 1 is two haploid cells, meiosis 2 begins without the chromosomes going through another round of DNA replication. Centrioles again duplicate and begin moving to opposite poles of each cell. In prophase 2, the sister chromatids condense while the spindle starts to form as the nuclear envelope disappears. In prometaphase 2, the sister chromatids attach to the spindle by kinetochore microtubules, with sister chromatids attached to opposite poles. The spindle aligns the sister chromatids along the metaphase plate during metaphase 2. During anaphase 2, sister chromatids separate and individual chromosomes move toward the poles. The entire process ends with telophase 2, as the chromosomes decondense and the nuclear envelope reforms. Cytokinesis occurs, and cleavage furrows separate the two daughter cells into 4 haploid daughter cells. The haploid daughter cells will specialize into gametes, either sperm or egg. These fuse in fertilization to form a zygote, which will grow into a child. The child receives half its chromosomes from its mother and half from its father. Because men and women produce millions of gametes, and the selection of gametes in fertilization is random, this contributes to genetic diversity. This explains why a child isn't identical to either parent. But why aren't siblings identical? The diversity comes from several sources. In prophase 1, non-sister chromatids can exchange DNA through crossing over, increasing the genetic diversity of individual chromatids. In addition to crossing over, in metaphase 1, the pairs of homologous chromosomes align independently along the metaphase plate and sort independently into the daughter cells. This process, called independent assortment, produces 4 genetically distinct haploid gametes. As the total number of chromosomes increases in an organism, the number of genetically distinct gametes increases by 2 to the nth power. This means an organism with n equals 3 can produce 8 unique gametes. For humans where n equals 23, there are 2 to the 23rd power unique gametes formed, or over 1 million different possible combinations. The combination of independent assortment, crossing over, and the random pairing of gametes during sexual reproduction increases genetic diversity and explains why a child will not look exactly like his or her parents or siblings.
Okay, so I hope you learned something from that video. Okay, it's very informational. It's very informative video explaining meiosis. Okay, as a process of reproduction or creating of cells. Okay, so Yan. So, to make a summary with that discussion, okay, in order for us to differentiate mitosis and meiosis, okay, we first identify the role. Okay, so the role of mitosis is that for growth, for cell reproduction, in a case of cell growth, okay, tissue repair, and etc. For, what do you call this? For meiosis, the purpose of that is for spermatogenesis or oogenesis, means creation of gametes or sperm and egg. Okay, so responsible for reproduction. Okay po? So, yan.